the organizers. I, I want to take the opportunity to give you a little bit of data on our attempts to understand what is driving the genetic diversity of PFS47 and its ortholog in P. vivax. So this is part of a studies trying to understand what are the main factors that allow Plasmodium ookinete to invade the mosquito midgut. And what we found a couple of years ago was that the Plasmodium parasite has a protein called PFS47 in the surface of female gametocytes and in the ookinete. And this PFS47 protein allows the parasite to evade the mosquito immunity, specifically the complement-like system of the parasite, which is very effective in some circumstances, destroying the Plasmodium parasite. This PFS47 appears to inhibit an apoptotic response of midgut cells that somehow change the parasite, apparently by protein nitration, and make the parasite visible to the complement-like system so that TEP1 can bind the parasite and label it for destruction. So this immune evasion by PFS47 is also connected with a very interesting genetic structure of its haplotypes around the world. Analyzing sequences of isolates from around the world, we found that the haplotypes of PFS47 that are in different continents, circulating in different continents, are different. Here are with different, I colored them in blue, the ones that are circulating in the New World, in Asia in green, and in Africa where there is larger diversity in orange, or tones of orange. This pattern is consistent with a parasite that evolved in Africa and was brought to other continents by human migration. Now, the different haplotypes in other parts of the world are also consistent with a selective process which we propose, that's our hypothesis, has been done by the different anophelines that are present in those regions. So we try to obtain evidence for a vector-driven parasite selection, and the hypothesis is that the immune system of the mosquito has been a major barrier for this parasite to adapt to other mosquitoes, and specifically that the target, one of the targets of this selection has been PFS47. So the initial evidence that we obtained for this evidence was studying 3 different anophelines that are evolutionarily very divergent. Anopheles albimanus, Anopheles gambi, and dirus. The evolutive distance between albimanus and gambi is 100 million years of separation when the 2 continents pull apart. This is in the middle of Cenozoic, when the mosquitoes were mainly biting dinosaurs before the diversification of mammals. So it's a long time. These mosquitoes are very, very distant. Now, dirus is distant from gambi probably about some 40 million years, still a large evolutive distance. So what we looked at first to obtain evidence of this selection of the parasite was just using falciparum isolates from different parts of the world and looking at the compatibility with these different mosquitoes. And we have, in this case, an infection with an putative African parasite, NF54. This one infects better the African gambi mosquito and less the Asian dirus and the New World albimanus. In a similar way, we tested a separate African isolate with the same result, more infection in the African mosquito, African anopheline. And when we looked at an Asian lines, we got better infection in the Asian Anopheles dirus, and a line from the New World, 7G8, we obtain relatively good infections in Anopheles albimanus, which cannot be infected well with the other isolates. So we think this is an evidence of compatibility, probably selection, and this is published. I just want to summarize the results. This lack of infection, especially in Anopheles albimanus, can be reverted by silencing the mosquito immune system, by disrupting the complement-like silencing LRIM1, for example. And it can also be reversed by exchanging which haplotype of PFS47 is in a given parasite. Taking an African line, we exchange it with an Asian haplotype of P47, and we changed the susceptibility toward Anopheles dirus or toward albimanus in the case of a haplotype from the New World. So we have this model in which we of course, we don't have all the evidence for it, but we think this works like a log-key system in which several PFS47 haplotypes have been selected by different anophelines around the world, and that's why we have this geographic pattern of PFS47. Now, so there are several questions that remain in this model. One of them is, what is driving the genetic diversity that we see of PFS47 in the African continent, which is very high? And the other one is, what is the lock in this system? What is the ligand in the mosquito that is interacting with PFS47? So with respect to the genetic diversity of PFS47 in Africa, we went, thanks to our collaborators from the Malaria Research Institute in Mali, the University of Bamako, Nafomon Sogoba and his team, we went to a small locality, and this is just a few miles, a few square miles, and they had been collecting mosquitoes and identifying the infected mosquitoes, a large collection, more than 10,000 mosquitoes. And so they were able to give us infected mosquitoes so that we could check what haplotypes of PFS47 are present in that locality and which ones are in the different mosquito species. Our hope was to analyze Anopheles gambiae and Anopheles funestus, which are distant apart. Due to the circumstances of the collection that year, we ended up having mostly Anopheles coluzi and gambiae sensu stricto. This is just a pattern of the collections of the two throughout that year, and this is a summary of what we found. We found 11 haplotypes in that small locality, so there's high diversity of PFS47 even in a small locality, that is Mali. And the most prevalent haplotypes, the three at the top, are in similar proportion in both Anopheles coluzi and Anopheles gambiae. That is, we didn't find a significant difference of what is circulating in both, which was not surprising to us because evolutionarily those two species are very, very close. Now, what is driving, again, the diversification of PFS47 in Africa? We are looking now at the human immune system. We and others have found that infected people in Africa do have antibodies against PFS47, and we have now evidence that antibodies raised in mouse have transmission blocking activity. So we think that at least in Africa, maybe the main driving force for PFS47 diversification is the human immune system. Now, we also looked at Plasmodium vivax, and the same pattern shows up in the haplotypes around the world circulating now, even more striking than falciparum. And thanks to a study that was done some years ago at NIH in collaboration, Cinzia Anzu's lab in collaboration with Lilian Gonzalez-Herón in Mexico, we were able to obtain samples from the southern part of Mexico in Chiapas, and their findings were really, really interesting, analyzing the population structure of Plasmodium vivax. They found that in the lowlands, where Anopheles albimanus is the main vector, there is a clear population of vivax circulating, and in the highlands, where Anopheles pseudopunctipenis is the main vector, is a different population. In fact, they were able to identify two different populations here, F1 and F2. So we were able to obtain these samples and sequence PV47 to see if if they are different, and they could be correlated with infection of the different vectors. And you can see here the experiments that they did with a large number of samples. This was done in Mexico, showing that the population of the lowlands infects better Anopheles albimanus, while the populations of the highlands infect better Anopheles pseudopuntipenis. So in summary, the result that we obtained was 6 different haplotypes of PVS47, and with a very clear difference between what is circulating in Pseudopuntipenis and Dirus. The haplotypes in Dirus are totally different from the ones in Albimanus. So at least here there is a correlation, but there is a population structure already there, so we don't know if this is causality. And now we are checking whether these haplotypes actually determine differential infection of Albimanus of Pseudopuntipenis in the lab, transforming Plasmodium falciparum with these vivax haplotypes. So now the last question that I want to address today: what is the nature of the ligand that is interacting with PFS47? The ligand, the mosquito ligand. So for this, we hypothesizing that it's a protein that may be important to trigger this apoptosis response in the midgut epithelia. We use a far western approach in which we ran midgut proteins, membrane or cytosol proteins, and then we probed it with recombinant PFS47, and then we detected this PFS47 if it bound to something in the western blot with an antibody. And essentially what we got in the membrane fraction of the midgut protein, we found mainly seeing a specific band which we were able to reproduce in a 2D gel and sequence the peptide. We have the identity of the protein, which is of unknown function. It's just, it has been annotated, but it's of unknown function. And when we silence that protein by RNAi, we're able to eliminate the binding that we see in the far western. So we think we got the right protein that we observe in the far western. And furthermore, this protein does have a phenotype when we silence it in the mosquito and we infect it with Plasmodium falciparum. That is, it decreases the level of infection. So we think that this protein may be a repressor of the apoptotic response. When we remove the repressor, now the apoptotic response is higher and destroys the parasite. We are in the middle of trying to figure out what is it connected to, the other proteins that interact with this apparently membrane protein. We have an IFA that is preliminary data. It seems to localize the protein toward the luminal side of the midgut epithelia. So we are now with a receptor in the middle of synthesizing recombinant receptors from the different anopheline species, so that we can try to reproduce the compatibility we see in the infections at the level of receptor and PFS47 in vitro. So we want to see if it really works that way, that a given PFS47 would be more would have more affinity to a given receptor according to the location of that receptor in a mosquito from Asia or from the New World. With that, I just want to conclude that as I told you, the genetic diversity of PFS47 in Africa may be driven in large part by the human immune system, not so much by the mosquito immune system. And that at least in vivax, we did find this separation between what is circulating in Anopheles albimanus and Pseudopuntipenis. And I forgot to remark that these two mosquitoes, Albimanus and Pseudopuntipenis, are evolutionarily apart. In fact, they are from two different subgenus, the Nisorhynchus in the case of Albimanus and Anopheles in the case of Pseudopuntipenis. So it makes sense that there would be more distance and more selection of PVS47. So we think we have a receptor, and there are a couple of implications for all of these that we think is important to mention. The first one is that what we see at the level of selection of PFS47 may be driving some of the population structure of Plasmodium falciparum and maybe Plasmodium vivax around the world. The selection by the vector hasn't been taken into account in a lot of the population genetic analysis of these parasites. And this is especially relevant for the case of dispersal of drug resistance, artemisinin resistance, since the artemisinin resistance marker that has been found, the culture 13, is closely located, physically closely located to PFS47, just around 150 kilobases. So it's genetically linked. Whatever selects PFS47 is also going to select calcium 13 and the other way around too. One conclusion of this is it may be very difficult for a parasite in Asia resistant to artemisinin to move to the New World because it would have a hard time infecting mosquitoes like Albimanus. So with that, I just want to acknowledge all the people at NIH that are involved in this work, especially the students and postdocs, and the group at Mali, the MRTC, Nafo Monsogoba, and Musa Keita. Thank you very much.
All right, we have time for one quick question.
Do you have any idea whether there are orthodox if you're not protein Gambia and the other species?
Yes. In fact, I showed very briefly a phylogenetic tree. Yes, there are orthologs, and in the case of Anopheles colucci and Anopheles gambiae, they are almost identical. Very, very high homology. And they are more distant between Anopheles albimanus, Nisorhynchus, and Anopheles gambiae.
Okay, thank you.